We tend to think of the lymphatic system as just the body's plumbing, dealing with excess fluid. But what if that's a massive underestimation?
Hmm.
Consider this.
Hmm.
This whole network, often invisible to us, plays an absolutely fundamental role in keeping the environment around our trillions of cells stable and healthy. It's a surprising idea with huge implications, isn't it?
It is, and it really underscores a point that often gets missed. Yes, fluid drainage is crucial, but the lymphatic systems work in maintaining that delicate balance of fluid and molecules in the spaces between cells, the intercellular liquid and extracellular matrix, that's essential for the basic survival and function of all our tissues.
Okay, let's really get into this. For this deep dive, we've compiled some fascinating material, stretching way back to historical observations, think ancient Greece, right up to the very latest in molecular research.
A huge span of time.
Exactly. Our mission for you today is to take this wealth of information and build a clear, comprehensive, and yes, even entertaining understanding of the lymphatic system. We're talking about its anatomy, how it actually works, and its critical role in keeping us well.
And, of course, what happens when things go wrong?
Right. Prepare for some genuine aha moments that might just change how you think about your own body.
Indeed. So, to begin with this broader perspective on what the lymphatic system actually does, it's vital to move beyond the image of simple drainage. This is an active system that's constantly working to maintain equilibrium within our tissues.
Right.
Think about the components involved. First, the network of vascular channels themselves. Then we have the lymphoid organs names you'll likely recognize, like lymph nodes, the spleen.
Pear patches in the gut, the thymus, tonsils, those ones.
Exactly those. And crucially, let's not forget the circulating cells within the lymph fluid. The lymphocytes and macrophages, which are key players in our immune surveillance.
Right, so it's not a static system. It's a dynamic network with cells on the move.
Yeah.
And it's carrying more than just water, isn't it? I was reading how even large molecules like immunoglobulins,
Antibodies, yeah.
antibodies, polypeptides, and even entire plasma protein complexes and signaling molecules called cytokines can move into the lymphatics.
And eventually return to the bloodstream. That sounds like a really significant role in monitoring what's going on in the body.
It absolutely is. It's a continuous circulation working alongside the bloodstream. While blood is rapidly delivering oxygen and nutrients to tissue fluid and lymph, carrying these immune cells and signaling molecules, well, they percolate through the lymphatic vessels and lymph nodes at a much more deliberate pace.
Slower, definitely.
What's truly fascinating here is to think of the lymphatic system as a dedicated, almost parallel loop within the extracellular space. It's essentially completing a circuit by collecting all those substances, fluid, large molecules, even cells that might escape or leak,
Right, the escapees from the thin-walled blood capillaries.
and diligently returning them to the main venous circulation.
That idea of escapees being reclaimed really makes it click. So let's step back in time for a moment. It's quite remarkable to think that figures like Hippocrates and Aristotle were already observing these vessels and nodes.
Yeah, centuries ago.
Even noting the intestinal lymphatics in animals that had recently eaten. That's centuries before we even had a proper understanding of blood circulation.
It is quite remarkable. And then if we fast forward to the 17th century, to Gasparo Acelius,
Ah, yes, Acelius.
his discovery of the chylous mesenteric lacteals, these milky white vessels in the intestines in a dog, happened around the same time that Harvey was meticulously mapping out blood circulation. That was a real turning point, wasn't it?
A huge turning point. It provided undeniable evidence that these lymphatic vessels were widespread and played a critical role in absorbing nutrients from the gut. These lymphatic absorbents, as they were sometimes called, essentially accompany veins almost everywhere in the body.
Almost everywhere. There are a couple of interesting exceptions, aren't there? Like the central nervous system, CNS,
Brain and spinal cord, yeah.
and the dense cortical bone. But even in those areas, the body has evolved its own ways of draining interstitial fluid.
Exactly. The CNS and cortical bone lack traditional lymphatic vessels, but they do possess specialized drainage mechanisms. Now, if we take a look at the macroscopic anatomy, the big picture, it's almost like a detailed map of the body's fluid highways.
Okay.
The majority of the lymph from the lower torso and the internal organs eventually enters the bloodstream via the thoracic duct,
That main big one.
which empties into the junction of the left subclavian and jugular veins in the neck. Meanwhile, the head, neck, and upper extremities have their own independent lymphatic pathways that drain into the central veins.
And it sounds like there's quite a bit of individual variation in these routes, a lot of interconnections. You mentioned that lymph from the heart and lungs often drains into the neck veins on the right side, for
Yeah,
example.
precisely. And there are lots of these subvariant pathways. Then you have the unique journey of lymph from the intestines. Because it's carrying cholesterol, fat-soluble vitamins, and those long-chain fats packaged as chylomicron.
Hey, fatty stuff.
It follows a different path through the retroperitoneum, that space behind the abdominal cavity, ultimately forming a collecting sac called the cisterna chyli,
It's cernically.
before joining with other visceral lymphatics to become the thoracic duct. What's particularly interesting is that lymph from the liver actually flows in the opposite direction to the blood in the portal vein
Countercurrent flow, wow.
and meets up with the intestinal lymph just before it enters the thoracic duct.
It's also interconnected and precisely orchestrated. And these anatomical variations aren't just textbook details, are they? You mentioned they can indirectly influence things like swelling in the limbs, peripheral edema.
Oh, definitely.
And they're definitely crucial for understanding those edema syndromes that involve the internal organs and that phenomenon of chylous reflux we'll discuss later.
They are indeed clinically very relevant. And thinking about those areas that lack traditional lymphatics, like the retina in the eye and the brain, it's remarkable how they've developed analogous systems. The canal of Schlemm in the eye, which drains aqueous humor,
Okay.
or the cerebrospinal fluid CSF drainage pathways in the brain, the subarachnoid villi, also known as Pacchionian bodies, and the glymphatic system.
The lymphatic system, right, it's been getting a lot of attention.
These all serve the essential function of draining interstitial fluid in those specific and critical environments. Think about that your brain has its own dedicated waste removal system operating in the fluid between cells. That fundamentally changes how we view brain health and disease.
The lymphatic system in the brain is particularly intriguing, isn't it? This idea of cerebrospinal fluid essentially washing through the brain tissue to clear out metabolic waste products.
It is, and it really highlights the fundamental necessity of interstitial fluid drainage in all tissues throughout the body. Even the brain, which was once thought to be entirely devoid of lymphatics, has these sophisticated mechanisms in place.
Amazing.
And when these lymphatic pathways are disrupted, you can observe significant clinical effects. For instance, if the cervical lymphatic trunks in the neck are interrupted, perhaps during a surgical procedure—
Like a neck dissection for cancer.
Exactly. It can lead to noticeable facial swelling and even a temporary neurological syndrome. Conversely, if you increase the fluid pressure within the brain, like infusing crystalloid into the cisterna magna,
Okay.
you can actually see an increase in lymph flow from the neck. It really underscores just how crucial an unimpeded circulatory system of interstitial fluid and lymph is for maintaining overall homeostasis, that stable internal environment we all rely on.
Okay, so we've got a sense of the big picture function and the major routes. Now let's zoom in at a different level of the development of this system, right from the embryonic stage. It sounds like there's been a long-standing debate about the fundamental question of where lymphatics actually originate,
Oh, a classic debate.
whether they sprout from existing blood vessels or arise independently.
That's a very accurate way to put it. The two primary theories have been lymphovasculogenesis, which proposes that new lymphatic vessels form from specialized endothelial precursor cells,
So starting from scratch, sort of.
and lymphangiogenesis, where they bud off from already existing blood vessels, typically veins. Sabin put forward the centrifugal theory, suggesting they grow outward from the central veins towards the periphery of the body.
Centrifugally, like center out.
Right. Kempmeyer, on the other hand, championed the centripetal theory, arguing for an independent origin of lymphatic vessels in peripheral tissues that later connects to the central venous system.
Centripetal, periphery in.
That's fascinating that even something so fundamental was debated for so long. It was. For decades.
And where does the current weight of evidence point? It sounds like more recent studies focusing on specific genes like PROX1 and VEGFR3. These are key molecular players in lymphatic development.
Absolutely crucial markers.
Have provided stronger support for that centrifugal theory, the sprouting from veins idea, but there still appears to be evidence for a contribution from the centripetal mechanism as well. It's not fully settled.
That's a very insightful summary. While the initial molecular signals did seem to heavily favor the centrifugal origin through sprouting from veins, more recent and detailed work has indeed revealed a significant contribution from mesenchymal lymphangioblasts, a type of embryonic cell in peripheral tissues.
Ah, so mixed picture then.
It now seems quite likely that both of these processes are involved, perhaps to varying degrees depending on the specific location in the body and the precise stage of development, ultimately leading to that essential connection between the lymphatic and blood vascular systems.
Fascinating. Now, if we shift our focus from development to the microscopic anatomy, what do these lymphatic vessels actually look like when we examine them up close? It sounds like they begin as these highly permeable capillaries nestled within the interstitial space,
Right, in the tissue fluid.
almost as if they're gently scooping up the surrounding fluid and larger molecules.
Precisely. And in certain organs, most notably the liver, they even appear to originate from these unique, non-endothelialized precapillary channels. What's truly remarkable is their exceptional permeability.
More permeable than blood capillaries.
Much more, generally speaking. Lymphatic capillaries are designed to readily allow even very large macromolecules, we're talking over 1,000 kilodaltons, much larger than what can typically cross the walls of most blood capillaries, to enter their lumen. They do share this high permeability characteristic with the sinusoidal blood capillaries found in the liver, however.
So they're specifically designed to let the big stuff in. But I can imagine it might be quite challenging to visually distinguish them from small blood vessels under a standard light microscope. You mentioned that they tend to have thinner walls, a more tortuous or winding appearance,
Yeah, irregular shapes sometimes.
a wider and more irregular lumen, and typically lack red blood cells.
That's generally the case, and those are helpful visual clues.
Ah.
However, without the aid of specific staining techniques, it can indeed be difficult to definitively differentiate them from small venules or capillaries.
Mm.
Over the years, various staining features have been employed, such as looking for factor VIII-related antigen, also known as von Willebrand factor, VWF.
A blood vessel marker, usually.
Right. However, the differences in staining intensity for these general endothelial markers tend to be more quantitative, less intense in lymphatic endothelium compared to blood vessel endothelium, rather than a clear-cut qualitative distinction. You couldn't always be sure.
That makes perfect sense. So the real breakthrough in studying these vessels must have come with the discovery and development of lymphatic-specific markers, right?
Mm-hmm.
Molecules like LYVE1 and PROX1. These must have been game changers for researchers.
Absolute game changers.
Mhm.
These lymphatic-specific markers have been instrumental in allowing us to definitively identify and distinguish lymphatic vessels from blood vessels in tissue samples. LYVE1, for instance, is a key marker for the initial collecting vessels and lymphatic capillaries.
Starting points.
And it seems to act almost like a welcome sign, playing a crucial role as an entry point for dendritic cells, important immune cells, into the lymphatic system.
Ah, so facilitating immune cell traffic, too.
Exactly. PROX1 is another vital one. It's a nuclear marker that is specifically expressed in lymphatic endothelial cells. Then we have podoplanin in mice and its human equivalent, which is recognized by the antibody D240.
D240, I've seen that used in pathology reports.
You probably have. This marker is particularly useful for distinguishing lymphatic endothelium, especially in the larger collecting lymphatics that may not always show strong staining for LYVE1. This has been invaluable in studying tumors and their interactions with the lymphatic system, particularly in the context of metastasis.
Crucial for cancer research.
Definitely. There are also other research markers, such as 5-nucleotidase and even some like VEGFR3 and neuropilin-2 that can show some cross-reactivity with blood vessels. But overall, we now have a much more precise and reliable toolkit to identify and study these previously somewhat elusive vessels.
And if we delve even deeper to the ultrastructural level using electron microscopy, what does that reveal about how these vessels are structurally adapted for their unique functions? You mentioned the presence of both open and closed endothelial junctions,
Right, looking really close up.
and that the basal lamina, the supporting layer, is often tenuous or even absent.
Right. At the ultrastructural level, lymphatic capillaries exhibit these unique endothelial cell junctions, which can be either relatively open or more closed with complex convolutions. This variability likely reflects their role in both allowing fluid and large molecules to enter and maintaining some level of structural integrity.
So adaptable junctions.
Exactly.
Mm.
Unlike blood capillaries, they possess a very thin or often absent basal lamina. And then there are these crucial anchoring filaments.
Anchoring filaments, what do they do?
They're essentially complex elastic fibers that tether the lymphatic endothelium to the surrounding interstitial fibrous gel matrix. This is a brilliant piece of design because when the pressure of the tissue fluid increases...
Like when swelling starts.
Precisely. These anchoring filaments pull on the lymphatic capillaries, effectively opening them wide and allowing for increased fluid uptake, whereas the adjacent blood capillaries might actually collapse under the same pressure.
That's an incredibly clever and almost self-regulating mechanism in response to fluid buildup.
Wow. You also mentioned the presence of different junctional structures within the lymphatic network button junctions found in the initial
Like
capillaries
little spot welds almost.
that then transition to more tightly sealed zipper junctions in the terminal lymphatics.
Yes, these distinct junctional morphologies likely reflect differing permeability properties and the increasing need for structural integrity and controlled flow as the lymphatic vessels mature and begin to actively transport lymph.
Makes sense. They get less leaky as they go along.
Pretty much. And while the very terminal lymphatic vessels lack a smooth muscle layer, their endothelial lining is rich in actin, a contractile protein that may contribute to their ability to modulate their shape and function.
Mm.
Further along the lymphatic pathway, in the larger collecting lymphatics and trunks, you see the development of prominent intraluminal bicuspid valves.
Valves, like in veins. Exactly, ensuring one-way flow. These valves create functional contractile segments known as lymphangions. These various microscopic features all work in concert to facilitate the efficient absorption and transport of those large molecules, cells, and any foreign agents that enter the interstitial space.
So as the saying goes, seeing is believing. How do we actually transition from these microscopic observations to being able to visualize the entire lymphatic system in a living organism?
It sounds like the earliest methods were quite rudimentary,
Very rudimentary.
essentially relying on observing those chyle-filled lacteals in the intestines.
That's indeed where our macroscopic understanding began, with those initial visual observations. Asellius's detailed color plate depicting these vessels was a landmark achievement. Then came more invasive techniques, such as injecting mercury into the lymphatic vessels of cadavers.
Mercury. Yikes. Yeah, different times. That allowed for more extensive mapping, followed by the detailed anatomical dissections performed by pioneers like Mascagni and Sappi. Later on, techniques employing silver nitrate and Prussian blue helped to better distinguish the lymphatic vessels from the surrounding blood vasculature.
Getting a bit more sophisticated.
And remarkably, that relatively simple yet effective method of vital dye injection, first pioneered by Hudak and McMaster in the 1930s, injecting blue dye, basically.
Blue dye under the skin.
That's the one. It's still utilized in certain clinical settings today.
It's amazing how some fundamental techniques can endure despite technological advancements. But then, of course, modern medical imaging technology stepped in. We're talking about lymphangiography, which involves injecting oily contrast agents directly into the lymphatic vessel,
A bit tricky to do, that one. Kinmonth pioneered it.
and whole-body lymphangiocintigraphy,
Right.
which uses radioactive tracers that are taken up by the lymphatics to provide a functional overview. These must have offered a much more comprehensive and dynamic view.
Absolutely. Lymphangiography provided detailed anatomical images of the lymphatic vessels themselves, allowing clinicians to identify blockages or abnormalities. Lymphangiocintigraphy, on the other hand, offered a whole-body perspective on the flow of lymph, although with somewhat limited spatial resolution.
Right, not super detailed pictures.
But it's been incredibly valuable for initial screening and for monitoring the progression of lymphatic disorders. And now we have hybrid imaging techniques like SPECT-CT,
SPECT-CT, combining modalities.
which combines the functional information obtained from scintigraphy with the high-resolution anatomical detail of computed tomography, CT scans, providing much improved sensitivity and localization of lymphatic issues.
And the field hasn't stood still, has it? It sounds like there's a whole array of more recent and increasingly sophisticated indirect lymphography techniques that utilize various types of particles and dyes, fluorescent particles, magnetic particles, infrared dyes.
ICG, indocyanin green, is a big one now.
Even immunoglobulin conjugates and microbubbles, which can then be detected using a wide range of imaging modalities, from fluorescent microscopy and optical imaging to CT, magnetic resonance imaging, MRI, ultrasound, and even photoacoustic imaging.
Yeah, the toolbox is getting really packed.
It really sounds like molecular imaging of the lymphatic system is rapidly becoming a reality, allowing us to see not just the structure, but also the function and even molecular characteristics of these vessels.
It is an incredibly exciting and rapidly evolving field, and the refinement of percutaneous MR lymphangiography, which involves the direct puncture of central lymphatic vessels, often guided by ultrasound or other imaging.
Directly accessing the deep lymphatics.
Has been a major advancement, offering both high-resolution visualization and the capability for targeted therapeutic interventions, such as injecting specialized glue to seal lymphatic leaks or placing endovascular stents to open up narrowed vessels.
Image-guided interventions, amazing.
And techniques like photoacoustic imaging and that ICG lymphography are also showing tremendous promise for precisely staging lymphatic disorders and for guiding supramicrosurgical procedures aimed at treating conditions like lymphedema.
Okay, so we can now visualize this intricate network, and we have a better understanding of what it's made of and how it develops. Now let's delve into the fundamental question of how this fluid, the lymph, actually moves throughout the body. We've mentioned Ernest Starling before in the context of fluid balance across capillary.
Starling's principles, yes.
He also had important insights into the lymphatic system, particularly regarding its role in returning proteins to the vascular system.
Indeed. Starling recognized that any protein that leaks out of the blood capillaries into the interstitial space essentially requires the lymphatic system to return it to the main circulatory system. You can't just reabsorb large proteins back into blood capillaries easily.
So it's the only way back for them.
Pretty much, and this really highlights the lymphatic system's critical role as a fine adjuster of the tissue microenvironment. It's constantly working to maintain the delicate balance of fluid, protein concentration, and osmotic pressure in the spaces surrounding our cells,
Keeping everything just right.
as well as playing a supportive role in nutrient absorption, waste removal at the local level, and even gas exchange.
So interstitial fluid and lymph are essentially the extracellular water in continuous motion within our tissues. And Starling's well-known principles concerning hydrostatic and protein osmotic pressure gradients across the walls of the microvasculature, that framework helps to explain how this continuous partitioning of fluid normally occurs.
Exactly. There's a continuous, albeit small, net filtration of fluid out of the blood capillaries into the surrounding tissues.
A slight leak, always happening.
And it's this slight excess fluid that the lymphatic system diligently picks up and returns to the bloodstream. It's a relatively slow flow compared to the rapid circulation of blood, amounting to only about 1.5 to 2.5 liters over a 24-hour period at rest.
Just a couple of liters a day compared to liters per minute for blood flow.
Right. This unidirectional flow of lymph is driven by a subtle hydrodynamic imbalance between filtration and reabsorption at the capillary level. What's fascinating is that when these Starling forces are disrupted, say in heart failure where venous pressure backs up, you tend to see the development of low-protein edema, where the fluid accumulation is watery. However, when the lymphatic flow itself is impeded or blocked, a condition known as lymphostasis, this leads to the accumulation of protein-rich fluid in the tissues.
Because those proteins can't get cleared out.
Exactly, and that's what we recognize as high-protein edema or lymphedema.
And the propulsion of this lymph fluid, it's not solely reliant on the pumping action of the heart, as is the case with blood circulation. You mentioned that the lymphatic vessels themselves have an intrinsic ability to contract.
That's the primary driving force behind lymph flow. The larger lymphatic trunks, and likely even the smaller collecting vessels, exhibit spontaneous rhythmic contractions of their individual segments, which are called lymphangions.
Lymphangions, these contractile units.
Right. These lymphangions are functionally defined by the presence of those intraluminal one-way valves that ensure unidirectional flow. There's also a secondary contribution from extrinsic haphazard forces, as they're sometimes called.
Haphazard. Like what?
Things like the pressure changes associated with breathing, the squeezing action of skeletal muscles during movement, and the pulsations of nearby arteries.
So movement helps a bit, but it's not the main engine.
Not the main engine. Interestingly, the contractile activity of these lymphangions is very responsive to the volume of lymph flowing through them. An increased rate of lymph formation leads to more frequent and more powerful contractions.
Like a sump pump turning on more often when there's more water.
That's a great analogy. It's somewhat analogous to Starling's law of the heart, where increased filling leads to stronger contractions.
It sounds like these lymphatic vessels aren't just passive conduits. They're actively pumping the lymph along. And you mentioned that this contractility is regulated by a variety of agents.
A whole cocktail of things.
Including the sympathetic nervous system, metabolites of arachidonic acid, and even signaling molecules produced by the lymphatic endothelial cells themselves.
That's right. The regulation of lymphatic contractility is a complex interplay of various signaling molecules and neural and local stimuli, and the sensitivity to these factors can even vary in different regions of the lymphatic system.
So it's fine-tuned locally.
It seems so. While we know that the larger lymphatic trunks possess smooth muscle cells responsible for these contractions, it's still an area of active investigation whether the very terminal lymphatic vessels and capillaries also have significant intrinsic contractile capabilities, maybe subtle ones.
And what about lymph nodes? We know they're absolutely critical for immune function, acting as filters and sites where immune cells interact. But you also pointed out that they could potentially act as a bottleneck.
That's a crucial point to consider pathologically. While lymph nodes are indeed central to our immune responses, swollen, fibrotic, scarred, or even atrophied shrunken lymph nodes can physically obstruct the flow of lymph
Right, blocking the pipe.
and contribute to the development of lymph stasis in the tissues upstream. Unlike some lower vertebrates, like frogs, which have specialized lymph hearts to actively pump lymph.
Frogs have lymph hearts. Cool.
They do. But mammals rely primarily on the intrinsic contractility of the lymphatic vessels themselves, and lymph nodes, while vital for immunity, can sometimes impede this flow.
So if we were to compare the flow dynamics in the lymphatic system with those in the venous and arterial systems, it's a fundamentally different picture. The heart is the primary pump driving flow in both veins and arteries.
Providing that main pressure gradient.
Whereas in the lymphatic system, it's this intrinsic segmental contraction of the lymphatic vessels themselves, often referred to as the propulsor lymphaticum. That is the main driving force.
Exactly. Mammalian lymphatic smooth muscle exhibits a rhythmic and coordinated pattern of beating, with contractile waves propagating rapidly along the lymphatic vessels, both from the tissues towards the lymph nodes and from the nodes towards the veins where the lymph re-enters the bloodstream.
Coordinated waves pushing it along.
Right. Those intraluminal one-way valves are absolutely essential in facilitating this efficient unidirectional transport of lymph. So the lymphatic system essentially functions as a series of interconnected micropumps that actively respond to changes in fluid volume and pressure.
Little pumps all along the network.
Pretty much. Even small increases in the pressure within the lymphatic vessels can lead to a significant increase in the frequency of these contractile events. And while the lymphatic system does exhibit a relatively higher resistance to flow compared to the venous system.
It's harder to push fluid through.
Somewhat, yes. But this intrinsic pumping capacity allows it to effectively overcome this resistance and maintain lymph flow.
And this lymphatic ejection force can be finely modulated by a whole host of factors, including local filling pressure, temperature, the activity of the sympathetic nervous system, various neurogenic stimuli, circulating hormones, and locally secreted cytokine signaling molecules. It's a very dynamic and responsive system.
Highly regulated.
You also touched on the idea of pacemaking activity within these lymphangions, which is likely driven by pressure-sensitive chloride ion channels located in the smooth muscle cells.
Yes, the rhythm seems to originate within the lymphangions themselves. It appears that pressure-sensitive chloride ion channels in the smooth muscle cell membranes play a key role in generating the electrical signals, the action potentials that initiate these contractions.
So the pressure itself triggers the pumping.
That seems to be a key part of it. Interestingly, more recent research using sophisticated computational modeling suggests that pacemaker activity is most likely to occur in the regions adjacent to the intraluminal valves.
Right near the valves. Why there?
This is thought to be due to the higher cell density and the specific patterns of cell-to-cell communication found in these areas, which are optimal for initiating and propagating those coordinated contractile waves. It's like the command center is right by the gate.
That's a fascinating level of detail. Now, there's a common perception that skeletal muscle movement directly enhances lymph return in much the same way it aids venous return, particularly in the legs, the muscle pump.
Ah, yes, the muscle pump concept.
But you mentioned that this isn't entirely accurate under normal physiological conditions for lymphatics.
That's a very common misconception. While external compression, such as from muscle contraction or massage, can certainly help to move lymph fluid when there is already an accumulation of edema.
When the system is overloaded.
Right. But under normal, healthy conditions, peripheral lymph flow is primarily driven by the spontaneous intrinsic contractions of the lymphatic vessels themselves. The fluid column in the peripheral lymphatic vessels is not as continuous as it is in veins. It's often interrupted by valves and empty segments, so external compression isn't as effective at directly propelling lymph forward.
Okay, so the intrinsic pump is the main player normally.
However, in situations of lymphatic obstruction and lymph stasis, when that fluid column does become more continuous and the intrinsic pumps might be struggling,
Then muscle action helps more.
then skeletal muscle action and external compression can indeed act as important auxiliary pumping mechanisms to help move the stagnant lymph fluid.
That makes sense. And gravity also has a differential effect on the lymphatic and venous systems, doesn't it? You mentioned that standing upright sharply increases venous pressure in the distal limbs,
Due to the weight of the blood column, yeah.
but it doesn't have the same direct effect on peripheral intralymphatic pressure, although the pulsation of nearby arteries might increase.
Correct. This difference in pressure response to gravity actually favors lymphatic fluid removal from dependent limbs. The lymphatic system becomes just as susceptible to the effects of gravity as the venous system, contributing to the persistent swelling. The beneficial pressure gradient is gone.
Ah, okay. So ultimately, this lymphatic transport route is the critical pathway for the movement of liquid, proteins, and those large macromolecules, especially the chylomicrac carrying dietary fats throughout the body.
Absolutely essential for all that transport.
And of course, we know it's also the primary route for the spread of cancer cells, lymphogenous metastasis.
Okay.
But you also highlighted the often underappreciated role of the lymphatic system in the initial uptake and clearance of microorganisms,
Right, dealing with infections at the source.
and in the broader context of immune defense.
Absolutely. While we often primarily associate the immune functions with the lymph nodes, the initial uptake and clearance of bacteria, viruses, and other microorganisms by the lymphatic vessels themselves in the tissues is a crucial first line of defense.
Before it even gets to the node.
Exactly. It happens in the early stages of infection and plays a vital role in triggering subsequent adaptive immune responses. There were some really insightful studies conducted in the mid-20th century that specifically investigated this lymphatic route in various viral and bacterial infections and the intricate dynamics of the resulting immune response.
Older research that's still relevant.
Very much so. And the groundbreaking work of Joseph Yaffe on the production and trafficking of lymphocytes and other lymphoid cells was absolutely fundamental to the development of modern immunology.
So he really focused on the cellular traffic.
He did. It's quite possible that revisiting some of those earlier investigations with the powerful tools and techniques we have today in molecular biology and imaging could yield new and important insights into a wide range of diseases.
Like what?
Well, conditions like AIDS, tuberculosis, parasitic disorders, and even the mechanisms of cancer spread. And as you mentioned earlier, there's even active speculation and research into the potential role of the lymphatic system in the pathogenesis and resolution of COVID-19.
It's truly amazing how our understanding of this system continues to evolve. And that brings us to the fascinating field of molecular lymphology, this relatively new era of research that's being driven by the incredible advances in molecular biology and the wealth of information generated by the Human Genome Project.
Right. The molecular toolkit really opened things up.
Lymphangiogenesis, the process of forming new lymphatic vessels, is a central focus here, as it's critical in both normal physiological processes and in the development of various pathological conditions.
Indeed. While uncontrolled and abnormal lymphatic vessel growth can lead to serious conditions like lymphangiosarcoma, the tightly regulated process of lymphatic endothelial cell proliferation and the formation of new lymphatic tubes, lymphangiogenesis, is absolutely essential for a vast array of physiological and pathological processes.
Like development, repair.
Development, tissue repair, immune responses, even the spread of cancer, unfortunately. For a long time, the primary focus in vascular biology was on angiogenesis, the growth of blood vessels.
Right, blood vessel growth got all the attention.
Lymphangiogenesis has only more recently garnered significant attention, even though the ability of the lymphatic system to regenerate and adapt is vital for maintaining health. And disorders involving lymphatic flow and lymphatic vessel growth are surprisingly common.
You mentioned some classic in vivo studies conducted by Clark and Clark, who were able to directly observe the outgrowth of lymphatic capillaries in living rabbit ear chambers,
Amazing experiments peering into a living ear.
providing direct visual evidence of lymphangiogenesis. And Pollinger and Fleury's work further elucidated the distinct connections that form between lymphatic vessels themselves, as well as between veins and arteries and arteries.
Those were indeed foundational studies that provided direct visual confirmation of the lymphatic vessel's remarkable capacity for growth and regeneration in a living organism. The experiments involving skin incisions that demonstrated the rapid formation of new lymphatic vessels within just a few days.
Wow, that fast.
And Bellman and Odin's detailed microlymphangiography studies documenting lymphatic regeneration through scar tissue, including the de novo appearance of functional valves, really underscore the dynamic nature of this system.
So it can grow back through scars.
It can. Subsequent studies utilizing electron microscopy further confirmed that these regenerated lymphatic vessels were able to restore their intricate ultrastructural features, allowing them to effectively resume their crucial functions.
And it sounds like lymphatic endothelial cells, while typically exhibiting a relatively low rate of turnover in healthy adult tissues,
Mm.
they don't divide much normally.
They're quiescent, yeah.
They can significantly ramp up their rate of proliferation and migration in response to injury or inflammation.
Yeah.
You also mentioned that they tend to be more active in fetal tissues and in the lymphatic vessels of internal organs compared to those in the peripheral tissues.
That's a correct assessment. And the ability to isolate and culture lymphatic endothelial cells in vitro in the lab from a variety of sources,
Growing them in a dish.
ranging from bovine mesenteric lymphatics to human tissue samples obtained from hygromas or lymphangiomas, as well as from canine and human thoracic ducts, and even from microvascular cultures, using those specific lymphatic markers like LYVE1 to identify and isolate the lymphatic cells. That was another critical step forward.
Allowed for controlled experiments.
Exactly. This allowed researchers to study the cellular and molecular mechanisms of lymphangiogenesis in a controlled environment, even demonstrating the spontaneous formation of lymphatic tubes in cyst-like structures in culture, or when these cells were embedded within a collagen matrix.
And this in vitro work really paved the way for our growing understanding of the genetic control of lymphangiogenesis, didn't it? It sounds like there's an increasing amount of evidence pointing to specific genes playing crucial roles.
Absolutely. We are increasingly recognizing that the process of lymphangiogenesis is under very tight genetic control, just like angiogenesis. For example, the observed association of lymphatic and cardiovascular abnormalities in individuals with Turner syndrome, a genetic condition involving the X chromosome,
Turner syndrome, right.
strongly suggests that genes located on either the X or Y chromosome are likely to be involved in the normal development of both the lymphatic and cardiovascular systems. Furthermore, various chromosomal aneuploidies, where there's an abnormal number of chromosomes, like Down syndrome, trisomy 21,
Okay.
and chromosomal rearrangements have been occasionally observed in association with conditions like cystic hygroma, lymphedema, and intestinal lymphangiectasia, further highlighting the genetic underpinnings of lymphatic development.
So chromosome issues can affect lymphatics.
They can. Both forward and reverse genetic approaches have been instrumental in identifying specific lymphangiogenesis genes, and we have now cataloged a number of hereditary lymphedema angiodysplasia syndromes, many of which follow an autosomal dominant pattern of inheritance.
Meaning just one copy of the faulty gene is enough.
Often, yes.
You gave some specific examples of these genetic links, such as mutations in the VEGFR3 gene in some patients with Milroy syndrome,
A type of congenital lymphedema.
mutations in SOX18 in hypotrichosis lymphedema telangiectasia syndrome, and mutations in FOXC2 in lymphedema distichiasis, LD syndrome. The fact that a mouse model with a mutation in the FOXC2 exhibits similar lymphatic abnormalities, that's powerful evidence.
It really is. Finding the same gene causing similar problems in mice and humans strongly points to its causal role. So these genetic links aren't just academic, are they? They're actually starting to explain why some people are predisposed to lymphatic issues.
Precisely. These genetic discoveries have significant clinical implications. Understanding the specific gene mutations involved in hereditary lymphedema can aid in diagnosis, genetic counseling, and potentially even the development of targeted therapies in the future.
Tailored treatments based on genetics.
Exactly. The FOXC2 mouse model, for example, allows researchers to study the precise mechanisms by which this gene affects lymphatic development and function, paving the way for potential therapeutic interventions. And the list of identified genes implicated in lymphatic disorders continues to grow,
It's expanding rapidly.
including genes like CCBE1, GJC2, PDPN14, GATA2, and many others, each associated with distinct types and presentations of lymphedema and related conditions. What's particularly striking is that these currently known mutations still only account for a relatively small fraction.
Just the tip of the iceberg, maybe?
Of the over 45 inherited disorders listed in the online Mendelian Inheritance in Man, OMEM, database that affect the lymphatic vasculature, suggesting that a much wider array of genes and genetic factors are likely involved.
It's a complex network genetically, too.
And it's not just the genes themselves, but also the various growth factors and inhibitory factors that modulate lymphatic growth, development, and remodeling. You mentioned key players like VEGF-C and VEGFD,
The vascular endothelial growth factors specific to lymphatics.
as well as the intricate interactions of the angiopoietin-1 and angiopoietin-2 signaling pathways.
Exactly. These molecular signaling pathways, along with insights gained from the study of transgenic, spontaneously occurring, and experimentally induced mutant mouse models, are absolutely crucial for unraveling the complex processes that govern lymphatic development and function.
Mouse models are key here.
Indispensable. These angiomodulatory factors play a critical role not only in normal lymphatic physiology, but also in understanding the pathogenesis of a diverse range of clinical conditions.
Like,
Well, congenital lymphatic malformations like cystic hygroma and Klippel-Trenaunay syndrome, as well as acquired disorders such as angiosarcoma, elephantiasis, and other conditions characterized by swelling, scarring, immunodysregulation, and even malnutrition.
okay, so we've explored the normal development and function at multiple levels. Now let's turn our attention to what happens when this intricate system breaks down. It seems the most obvious consequence is lymphedema, which you described as insufficient lymphatic drainage, leading to a low output failure of the system.
Right, the pump isn't keeping up with the load.
But you also pointed out that experimentally reproducing persistent peripheral lymphedema has been surprisingly challenging. Why is that? Is the system just really resilient?
That's a very insightful question. Despite the seemingly straightforward idea that blocking lymph flow should lead to swelling, initial experimental attempts to induce chronic peripheral lymphedema through surgical removal or sclerosis, scarring of lymphatic vessels often failed.
The body just compensated.
Largely, yes.
Mm.
This was attributed to the lymphatic system's remarkable ability to regenerate and to develop auxiliary drainage pathways, including the formation of new lymphatic venous shunts. This led to the long-held theory that a secondary insult, most notably bacterial infection, lymphangiogenesis,
Infection on top of the blockage.
was often necessary to disrupt the microvasculature further and promote the more persistent lymphatic obliterations seen in chronic lymphedema. This idea was consistent with the delayed onset of lymphedema sometimes observed after procedures like mastectomy or groin dissection, and with the severe deformities seen in tropical filarial lymphedema, where repeated infections are a hallmark.
But it turns out that non-pitting brawny edema, the kind we typically associate with lymphedema, can indeed arise solely from lymph stasis itself, without needing infection. You mentioned Drinker's experiments with silica particles.
Exactly. Those studies, along with detailed observations in experimental animal models involving circumferential lymphatic transection, revealed a more nuanced picture. These models often showed an initial phase of acute edema that might then subside,
The swelling goes down temporarily.
leading to a period of latent lymphedema where there's no obvious swelling. However, this latent phase can eventually be followed by the reappearance and persistence of chronic lymphedema. Interestingly, factors like radiation exposure were found to exacerbate this progression.
Radiation makes it worse.
And similar sequences have been observed in experimental models of filarial infection. What's particularly important to note is that even during that latent phase, when there's no visible edema, lymphographic imaging can reveal ongoing and progressive damage to the lymphatic vessels themselves.
Damage you can't see on the surface.
Right. Increasing tortuosity and dilation leading to lymphangiogenesis, valve incompetence, and dermal backflow of lymph. Microscopic examination during this phase can also show cellular infiltration, destruction of the lymphatic vessel walls, and the deposition of collagen, scar tissue,
Scarring around the vessels.
eventually leading to a complete loss of distinct lymphatic structure in affected areas. This strongly suggests that extensive impairment of lymph drainage is indeed sufficient to cause chronic lymphedema.
So enough damage, even without infection, will do it eventually.
It appears so. The often long and unpredictable interval between the initial lymphatic disruption, such as after cancer surgery, and the development of refractory lymphedema can likely be attributed to this progressive, sometimes subclinical lymphatic damage.
That makes the analogy you drew earlier with deep venous occlusive disease leading to post-thrombotic syndrome even more relevant. Initial damage leading to chronic changes later on.
Precisely. Just as the absence or obliteration of venous valves and chronic venous obstruction and deep vein thrombosis can lead to the characteristic skin changes of post-thrombotic syndrome, the absence or obliteration of lymphatic vessels and lymphatic valve incompetence can lead to the distinct skin changes associated with post-lymphangitic syndrome,
Such as
such as the stemmer sign, inability to pinch the skin at the base of the second toe, warty overgrowths, papillomatosis, and that firm, leathery induration of the tissues.
And beyond the more typical presentations of lymphedema, there's a whole spectrum of other, often less well understood and more challenging to treat lymphangiodysplasias. You mentioned that these can sometimes involve both lymphatic and blood vessels.
Mixed malformations, yeah.
And can range in presentation from simple birthmarks to complex syndromes with mixed vascular components and even a potential for malignant transformation into lymphangiosarcoma.
That's a very accurate depiction. These are a diverse group of congenital and acquired disorders affecting the lymphatic system, often with overlapping features involving the blood vasculature as well. Diagnosing and managing these conditions often requires a multidisciplinary approach and the use of various diagnostic modalities.
What kind of tests help?
These range from simple clinical assessments like circumferential limb measurements to more sophisticated techniques such as bioimpedance analysis, tonometry to assess tissue stiffness, CT and MRI scans for detailed anatomical imaging, DXO scans to assess body composition changes, and ultrasound.
Lots of imaging options.
Right. However, whole-body lymphangiocintigraphy remains a crucial tool and is often considered the current gold standard for evaluating the overall function of the lymphatic system. More recently, memoir lymphangiography is playing an increasingly important role in providing detailed imaging of the central lymphatic channels and guiding targeted interventions.
The MRI for the deep stuff.
While ICG lymphography and photoacoustic imaging are proving valuable for assessing local lymphatic pathology, particularly in the context of surgical planning.
You also discussed the concepts of chylous and nonchylous reflux, this retrograde or backward flow of lymph, and how the classification depends on the origin of the lymph fluid.
Exactly. Chylous means it contains chyle, that fatty lymph from the intestines. Nonchylous is lymph from elsewhere. And because the lymphatic system is the sole absorptive pathway for cholesterol and long-chain triglycerides from the intestines,
Only way to absorb those fats.
any significant dysfunction of the mesenteric lacteals, the cisterna chyli, or the thoracic duct can lead to a range of clinical conditions characterized by the leakage of this lipid-rich lymph known as chyle.
So leaky chyle causes problems, like what?
These conditions include chylothorax, chyle in the chest cavity, chylocystitis, chyle in the abdominal cavity, chyluria, chyle in the urine, chylometrorrhagia, chyle in the uterus, and protein-losing enteropathy, where protein-rich lymph leaks into the intestines.
Oh, chyle can leak into lots of places.
It can. Furthermore, a high-grade blockage of intestinal lymph flow can lead to a backup of pressure, resulting in the dilation of peripheral lymphatic vessels and subsequent reflux of chyle-rich lymph into other tissues,
Backflow into legs or pelvis.
such as the pelvic or lower extremity tissues, causing chylous vesicles or chylodema. In very severe cases, this reflux can even extend into the bronchial tree, leading to the expectoration of chylous fluid,
Coughing up chyle.
a rare condition known as chyloptysis. Yes.
And it sounds like researchers are even using animal models to better understand these complex reflux syndromes. You mentioned mouse models.
Yes, these animal models, such as the angiopoietin 2 knockout mouse, as well as those with radiation-induced or spontaneous lymphatic abnormalities, provide valuable tools for studying the mechanisms underlying chylous reflux and for testing potential therapeutic strategies.
Helps figure out treatments.
Exactly. And what's increasingly important in clinical practice is that dynamic imaging techniques are now available that can help to identify the specific source, timing, and location of lymph reflux in patients of all age groups.
Pinpointing the leak.
Precisely.
You also highlighted the concerning increase in life-threatening chylous reflux in children who have complex congenital cardiac anomalies. You mentioned this is often related to elevated central venous pressure.
That's a significant clinical challenge we're seeing more frequently. The increased pressure in the central veins in these children can severely impede the normal drainage of lymph through the thoracic duct into the veins.
So the heart problem causes a plumbing backup for the lymph.
Essentially, yes. Consequently, there are ongoing efforts to develop better ways to image and correct this lymphatic overload and obstruction, often involving surgical interventions or endovascular procedures like stenting to decompress the thoracic duct,
Trying to relieve the pressure.
by bypassing the area of resistance at the cervical junction, or by addressing the underlying issue of excessive lymph formation due to the elevated venous or portal pressure.
Now, another devastating sequela of peripheral lymphedema is the increased susceptibility to recurrent cellulitis, a bacterial infection of the skin and underlying tissues. You mentioned it's most commonly caused by streptococcal bacteria,
Strep is the usual culprit, yeah. Erysipelas.
but more fulminant and severe infections with other organisms can also occur, and individuals with lymphedema seem to be particularly vulnerable compared to those with other types of edema.
They are indeed at a significantly higher risk. The exact reasons for this increased susceptibility are still being investigated, but several factors are likely at play.
What are the theories?
These include potential defects in local complement activation, a degree of immunodysregulation that has been observed in both canine and human filarial lymphedema, and evidence of dampened function in monocytes and macrophages.
Immune cells not working right in the swollen area.
That seems to be part of it. Other alternative hypotheses include the potential depopulation of regional lymph nodes due to the accumulation of fat and scar tissue, as well as a possible deficiency in the protease activity of macrophages within the lymphodematous tissues.
So, multiple potential immune problems.
It's likely a combination. It's often observed that the onset of lymphedema itself can be triggered by an episode of infection or injury in a limb that already has some degree of underlying lymphatic compromise,
The infection tips it over the edge.
initiating a vicious cycle of inflammation, lymphatic damage, and increased susceptibility to further infections, eventually leading to severe limb deformity and, in rare cases, the development of vascular malignancy.
That vicious cycle sounds terrible.
It is. While prophylactic antibiotic or antifungal strategies are used in filarial endemic regions to reduce the frequency of these infections, there isn't strong evidence to support the routine use of such prophylaxis in Western or non-tropical countries for lymphedema.
So just taking antibiotics all the time isn't standard.
Not generally recommended without recurrent infections, it's thought that effective lymphedema management itself, through measures like compression therapy and exercise, may be the best way to reduce the overall risk of infection by reducing the swelling and stasis.
Controlling the lymphedema helps prevent infection. Makes sense. And it's not just the fluid accumulation and the risk of infection that characterize lymphedema. You also highlighted that progressive interstitial fibrosis, the buildup of connective tissue, is another key feature.
That's a crucial distinction. The chronic presence of high-protein edema, as seen in lymphedema, appears to be a major driver of this excessive fibrous tissue proliferation, and also the increased accumulation of adipocytes, or fat cells, in the affected tissues.
More fat buildup, too.
Yes, although the precise sequence of these events is still being elucidated. Several potential contributing factors have been identified.
What drives the scarring and fat?
Well, altered production of various cytokines and growth factors, perturbed local immunoreactivity, the accumulation of abnormal protein complexes, which may even include growth factors themselves, proliferation of mast cells with the release of histamine, alterations in the sol-gel state of the extracellular matrix, and activation of the complement cascade.
A whole inflammatory soup going on?
Pretty much. These factors can collectively promote the infiltration of chronic inflammatory cells into the tissues. One important mechanism involves the accumulation of fibrin and cell-binding fibronectins, which act as a scaffolding for the migration of fibroblasts and the subsequent deposition of collagen,
The building blocks of scar tissue?
the main structural protein in scar tissue. In lymphedema, it's thought that lymph stasis impairs the activity of proteasome enzymes that normally break down these proteins, leading to a net accumulation of collagen and diffuse scarring.
So the cleanup crew can't keep up.
That's a good way to put it.
You also touched on the long-recognized link between the lymphatic system and the absorption and deposition of fat. You mentioned that lymphodematous limbs tend to accumulate more fat and lose it at a slower rate.
That's right. There's this intriguing hypothesis stemming from studies on mice with reduced levels of the PROX1 protein, suggesting that lymph itself might directly stimulate the differentiation and growth of fat cells.
The stagnant lymph promotes fat growth.
That's the idea, supported by more recent studies on mouse tails where experimentally induced lymph stasis led to increased fat tissue development, adipogenesis.
Wow. How does that apply to humans?
While the precise mechanisms in humans are still being worked out, there is also evidence suggesting that static lymph can influence localized adipocyte hypertrophy, meaning the existing fat cells become larger. It's important to consider that adipocytes in different locations behave differently.
Fat cells aren't all the same.
Right. And they likely have different cytokine growth factor profiles. Interestingly, in vitro studies have shown that pre-itocytes, the precursor cells to mature fat cells, can stimulate the growth of lymphatic endothelial cells.
Fat cells stimulating lymphatics.
And the addition of lymph fluid to pre-adipocyte cultures can actually promote their differentiation into mature adipocytes. So there's a complex interplay.
A two-way street, potentially.
Could be. This permanent fat accumulation in lymphodematous tissues is a significant factor that often limits the effectiveness of traditional physical therapies, like compression and massage.
You can't squeeze out fat.
Exactly. Which is why liposuction is sometimes considered as a therapeutic approach in carefully selected patients with advanced lymphedema where fat is a major component.
Finally, you discussed the rare but very serious sequela of long-standing chronic lymphedema, the development of lymphangiosarcoma or angiosarcoma,
A malignant cancer of the vessels.
an aggressive cancer of the lymphatic or blood vessels. Historically, this was perhaps best known as Stewart-Treves syndrome, which was associated with lymphedema following mastectomy and radiation therapy for breast cancer.
Right, the classic association.
However, it's now recognized to occur in other types of secondary lymphedema and even in cases of primary congenital lymphedema.
Yes, this is a devastating but thankfully rare complication. While initially most strongly linked to post-mastectomy lymphedema, we now understand that it can arise in the context of various forms of chronic lymphedema, both secondary and primary.
So not just after breast cancer treatment.
And it can develop many years after the onset of the swelling, sometimes even in the absence of prior radiation therapy. This suggests that the chronic lymphedema process itself likely plays a primary causal role in its development, possibly due to the long-standing inflammation and altered tissue environment creating a niche for malignancy.
The chronic inflammation might drive the cancer.
That's a leading hypothesis. It's also worth noting the connection to Kaposi sarcoma, another vascular tumor that is associated with a viral infection, human herpes virus 8, and is more commonly seen in the context of AIDS, which may potentially originate from virally transformed lymphatic endothelial cells.
Kaposi sarcoma involves lymphatics too.
Potentially, yes. These observations, along with the existence of other vasoproliferative and lymphologic syndromes, point towards a potential underlying role of immunodysregulation in a range of these conditions.
Like what other conditions?
Things like hemolymphangioma, Klippel-Trenaunay syndrome, Gorham-Stout disease massive, osteolysolysis lymphatic malformation association, MLA, angiofollicular hyperplasia, Castleman disease, lymphangioliomyomatosis, LAM, Kaposiform hemangioendotheliomatosis. It's a complex group.
And even tumor growth and spread.
Right. Tumor lymphangiogenesis and lymphangitic metastasis. Excitingly, specific gene mutations in the RAS-MAPK signaling pathway have been identified as underlying some of these lymphatic malformations.
We're finding the genetic drivers.
And there have been reported successes with directed gene therapy or targeted drug therapy using inhibitors like sirolimus in certain cases. In many of these complex lymphatic malformations, lymphatic obstruction plays a significant role, and central lymphatic decompression procedures, alongside tumor debulking and control of Sheilus leaks, often achieved through image-guided sclerosis or gluing, are important aspects of management.
So managing the plumbing is key even in these complex cases.
Often, yes. Finally, while we've extensively studied tumor angiogenesis, the formation of new blood vessels in tumors, a field pioneered by Judah Folkman tumor lymphangiogenesis, which was initially questioned.
People didn't think tumors grew new lymphatics.
For a while it was debated, but it's now recognized as a crucial area of research. There's significant overlap and even potential interchangeability in the cells that give rise to blood and lymphatic vessels during tumor development.
So it's linked to metastasis.
Highly linked. And the active pursuit of anti-lymphangiogenic pharmaceuticals by biotech companies for cancer treatment highlights the importance of this field. Beyond cancer, there are other lymphangiogenic disorders where the specific underlying disturbances aren't fully understood, but likely involve dysregulated lymphatic growth and remodeling.
Where things go wrong, but we don't know exactly why yet.
In the context of inflammation, infection, and immunodysregulation, these include primary disorders like lymphangioliomatosis, lymphangiectasia, and lymphangiomas, where the lymphatic involvement is clear, but the precise molecular and cellular details are still being actively investigated.
This has been a truly deep and fascinating exploration into a system that is so much more complex and vitally important than many of us likely ever consider.
It really is often overlooked.
From its fundamental role in maintaining the very environment our cells live in, to its intricate anatomy and physiology, the groundbreaking insights we're gaining from molecular lymphology and genetics, and the multifaceted nature of its disorders. The lymphatic system is clearly an unsung hero.
Definitely playing a major part in our overall health and well-being.
Absolutely. Through this deep dive, you now have a much greater appreciation for this often overlooked network and its profound significance.
Hopefully, yes. It's a critical player.
So, as we bring this discussion to a close, let's leave you with this provocative thought. Consider the incredible amount of ongoing research that continues to unlock the secrets of the lymphatic system. Think about the potential future of lymphatic medicine,
Huge potential there.
particularly in the development of targeted therapies for challenging conditions like lymphedema and cancer, and the exciting possibilities of engineering new ways to support or even bioengineer this absolutely crucial network within our bodies.
Regenerative medicine for lymphatics.
Who knows? It really makes you wonder about the far-reaching implications of this intricate system for your own health and for the future of medical advancements.